Hi again, everyone. Welcome to the discussion on Chapter 4. Pretty good chance we're going to break this up into two parts. I'm not sure yet. We'll see how it goes. I've got 41 slides, which means if I'm going a minute or two on each one, that's probably going to put us probably into the two-part phase. So hang tight. So about this chapter, the fundamental goal here is to delineate on a physiologic level this process of taking this energy inputs and converting it into things that, for instance, allow you to run a 100-yard dash or to get up out of your chair and drive to the university or walk to class, or turning this into the signals that are comprise our thoughts, emotions, and psyche or any number of things. But there's also the process of, as we do that, heat loss, we're losing energy in this process, but we're also having to eliminate materials that are no longer serve that purpose in the form of waste. We talk about metabolism and so forth. So it's about energy flow, but we're taking it to the next level because so much of us understand this stuff intrinsically. Now it's time to take it to the next level. So we're going to talk about energy and biologic systems a little bit, potential and kinetic energy and so forth. Where the meat of this lecture really goes is in, like, for instance, the anatomy of a chemical reaction. So we're going to take, those of you who haven't had a ton of chemistry, you might have seen some of this. A lot of this is to help set up those folks who are going to be taking standardized tests or thinking about graduate school. But we're just going to touch on it to help you have a fundamental understanding of what energy flow really means and why we need enzymes. Metabolism, we're going to talk about it writ large for sure, just a little bit. But where we're really going to delve into is how metabolism is controlled at the individual reaction level and not those big all-encompassing flow charts that you see from time to time that we call quote unquote metabolism. And the real big side of this is enzymes. So the heart of our lecture today is going to be about enzymes. These guys help control metabolism and help control chemical reactions. Well, then the question is, how do we control the controllers? If these guys control, how do we control them, if you know what I mean? So that's the fundamental question for our enzyme section of the lecture today. So a little bit about energy. You have heard some of this before, and you might have learned some of the Newton's laws and that sort of thing. We talk about energy as the capacity to do work. Well, what does that really mean? Right? That's sort of ambiguous to me. It's really about mass and the capacity to change the motion of that mass. If you're talking about this, like an object flying through the air, or are you talking about how that object, how hot it is, or whether that object is releasing light, those are all different forms of motion, just different levels of where that motion is occurring. And so I would say energy is the capacity to change the motion of mass. I think that's a better way of putting it. Hopefully you'll be able to see that as I do. I hope so. But in any case, let's talk about different kinds of work that capacity, that energy has the ability to do. Making and breaking of chemical bonds. When we make a chemical bond, what we're doing is we're storing energy for potentially later use. So we make a chemical bond. We might have to invest energy to make that bond if it's not really used to release energy. It might be for an infrastructure. But nevertheless, energy is stored when we make a bond. And energy is released to do other things, changing the motion of what's involved when we break a chemical bond. We also have transport work, that is, like, for instance, moving ions from the intracellular compartment to the extracellular or vice versa, moving molecules, and that can be across tissues or through cells, you name it. Even larger particles in some cases. Like, for instance, transport work might involve muscular motion and contraction to cause what we call bulk flow. Very useful transport work for creating concentration gradients. And here's an example of where we're converting energy. We're taking bulk motion, moving an ion, and that sets up a gradient for a different kind of motion. And then we have mechanical work. For instance, muscular contraction, moving organelles, changing cell shape, flagella, and so forth. Contracting muscles. Again, what we might do is contract muscles in order to move blood, for instance, and that creates a concentration gradient out of the tissue that allows for exchange. Now, I might be talking really fast here. I hope I'm not getting ahead of what you can envision, but these are kind of the three big things that we work on in biological systems. We have two primary forms. We have potential energy, and here are some examples, and kinetic energy. Over here are some examples. Potential energy is stored energy, either by virtue of the chemical bonds that it contains, the concentration gradients that exist, or in some cases the position of where that molecule or item is an example of potential energy. We also, but it has to be converted. You can't just, potential energy is not enough. It has to be transformed into a form of kinetic energy in order for the process to be completed. So kinetic energy is motion. It's either a bulk motion, like in the case of your heart squeezing blood through your blood vessels, or it can be the heat of a molecule, the molecular vibration, or even the light emission. In some cases, are forms of kinetic energy. Always involves some kind of motion. So here's just some couple of fun little stick figures. He's investing chemical energy in order to create potential energy in this object by virtue of its position in a gravitational field. And as soon as we release that object, it will obviously fall down to a lower energy state. It's releasing energy in the form of heat, okay? But in biologic systems, we often will harness that. So as this is trying to go to a lower energy state, what we will do is trap some of that kinetic energy and use it for other purposes. We call that phenomenon coupling, where we couple an energy releasing reaction with something that requires energy. And examples here, I've just got some pictures of some things where we're converting the chemical energy inside the muscles of these horses into mechanical energy that we can then use to do some kind of work. If you are new to UIndy or new to Central Indiana, this is actually a wonderful place to go if you have a couple of days off. It's Spring Mill State Park. They have a lovely example of where they're coupling water moving down its from potential energy by virtue of potential energy in a gravitational field into motion that grinds up corn. They have really, really great cornmeal there. This discussion about energy flow and conversion of energy is fundamentally one of the topic of bioenergetics. And there's a lot of interest in this because if you understand where the energy is, you understand when things are going to happen and when they aren't. And you can take this to a mathematical level. And so you can actually make arguments about whether a process is occurring or not based on the amount of energy that you know is involved. Where we're going to go now is into the idea of chemical reactions, where you have something that's reacting to become something else. And the idea that that will occur at a given rate based on how much energy is, potential energy is available and how much of that energy is released either as a byproduct or how much of that energy drives something else in the reaction. That's a key thing. How much energy is it going to take to get this started? In biologic systems, you're talking about fine shades of difference between the reactants and the products. There's a relatively small changes in energy. And so they may not be necessarily spontaneous under the given conditions. And so what we have to do is we have to either modulate how much energy is there, like in the form of temperature, or find some way to make the reaction more spontaneous. The more activation energy is required, the less spontaneous that reaction will be. And there are some reactions that may not occur for billions and billions of years just because there usually isn't enough energy to cause it to happen. For instance, let's say you have a sheet of paper in front of you, and if you have one, just grab it. You hear mine here. You can turn this sheet of paper into carbon dioxide very, very rapidly by simply lighting a match. And the next thing you know, the paper is on fire, and a lot of it's being converted into carbon dioxide. Some of it's being converted into ash. Happens relatively quickly. That can happen spontaneously as well. If we take this sheet of paper and we put it in put it somewhere for 1,000 years, when we come back, a lot of that paper has spontaneously turned into carbon dioxide. That's why old newspapers turn brown and age and so forth and get very, very brittle, because the material is literally turning into carbon dioxide. It's not a very spontaneous reaction. By adding activation energy in the form of a cigarette lighter, you can speed up the reaction quite a bit and make it spontaneous. That's the point of enzymes. That's the point of enzymes. Enzymes lower the need for activation energy and make the reaction more spontaneous under physiologic conditions. We oxidize glucose all the time. If you had a pile of glucose sugar on your table and you lit it on fire, it would burn at somewhere between 500 and 750 degrees. Well, we don't burst into flames. We oxidize that glucose in a controlled way so that we only need our temperatures to be somewhere around 98.6 degrees Fahrenheit, or even a little less than that. It's another way of indicating temperature sensitivity, is how much activation energy is required. Then we have this other concept, and this is probably the one time we'll talk about this other than endergonic and exergonic. But it does show up on standardized tests. We call this the delta G, the Gibbs free energy. How much energy is released into the environment because of the reaction, or how much energy does it absorb? When it releases, it has a negative delta G. When it absorbs, it has a positive delta G. But that's really all I'm going to talk about Gibbs free energy. I will show it to you graphically, though, going forward. And here's just an example of what I mean. A diamond is pure carbon, and it can exist for literally billions and billions of years without turning into carbon dioxide. But if you put enough energy in, you have reactants at the right concentrations, in this case liquid oxygen coupled with lots and lots of heat, you can cause a diamond to literally catch on fire and disintegrate in a matter of seconds. I watched a big fat diamond when I was in high school being burned this way on a video. Pretty amazing stuff. If you have enough activation energy, even very non-spontaneous reactions can become spontaneous. So here is an anatomy of a chemical reaction, with energy state being on the y-axis. In this case, we have time on the x-axis. And here's that activation energy. So we have the reactant here, and it may be very, very stable at this level. It contains potential energy, but we have to invest more energy to get over that requirement for energy input. Once it gets over that requirement, then the reaction becomes spontaneous, and what we see is that big ball breaking up into smaller balls, ultimately at a lower energy level. They have lost energy in the form of heat and molecular vibration. This change from the starting point over here to the final point over here is that delta G that I was talking about. In this case, we've gone from a higher place to a lower one, so this would be a negative delta G, because that energy that was in these things has been lost to the environment or kinetic motion. But what an enzyme will do, an enzyme and catalysts writ large, enzymes are biologic catalysts. Enzymes lower the activation energy requirement so that this reaction of taking this reactant and turning it into products becomes spontaneous at physiologic temperature. That's the key. By lowering the need for activation energy, you make a reaction more sensitive to the energy that's already there. We have energy stored in the chemical bonds here, but we also have the object has temperature, right? The environment has temperature, and temperature is energy. So if we make this activation one flatter and flatter, That means that the energy in the environment may be sufficient to trigger that spontaneity of a chemical reaction. Then what we're going to do in biologic systems is control reactions using those enzymes. And we're also going to control reactions by controlling the enzymes that control the reactions. So it becomes a sort of a sequential. So we got 2 or 3 balls in the air now, right? And so enzymes are incredibly important for facilitating reactions in physiologic conditions by lowering the activation energy requirement. But there's another role for enzymes in biologic systems, and that is to use that energy for something else that requires energy. We call that phenomenon coupling. Our grist mills were examples of coupling. We're releasing the kinetic energy from the stored up gravitational field with water, turning it into kinetic energy in the mill in order to break chemical bonds in corn seeds. Okay, that's coupling. We are releasing energy, and enzymes will facilitate the transfer of that energy to something else that requires energy in order to go forward. So we're coupling spontaneous ones, exergonic reactions. Exergonic reactions release energy, and we're coupling it to endergonic reactions that will not occur without energy input. So enzymes lower activation energy. Enzymes and enzyme complexes work together to couple endergonic and exergonic reactions. And that's what this figure is showing you here. We've got an exergonic reaction on the top, and we've got an endergonic reaction on the bottom. Negative delta G up here, positive delta G down here. So I've got some terms for you here. Now we use those enzymes, we couple those reactions like we have right here, and we call this process metabolism. Now there are different meanings behind metabolism. They can be all the chemical reactions that take place in an organism, or we can talk about something being metabolized. We can talk about individual metabolic reactions. For instance, we have catabolic reactions, things that break chemical bonds and release energy. Generally another word for exergonic. Then we have anabolic reactions, things that require energy in order to go forward. Another name for endergonic. When we are doing anabolic, if you ever heard the phrase anabolic steroids, we're building up our muscles. We're going to need to release energy in order for that buildup to occur. And that's coupling. So catabolism is breaking down, lowering energy state. The most common reaction for catabolism is hydrolysis, where we use water to break up a polymer like a peptide. We break a peptide into 2 smaller pieces. What water does is an H goes on one and an OH goes on the other, and you now have 2 smaller pieces using hydrolysis. The other side of it is when we're putting things together, like making polymers, we call that dehydration synthesis, because what happens is when you put those 2 smaller pieces together, like if let's say we're reversing this, you're going to end up with a water molecule out of those pieces. An OH from one molecule and an H from another creates H2O. Dehydration synthesis. And many times these things may involve multiple steps. This is kind of getting us back to this all chemical reaction thing. If we are, for instance, metabolizing glucose, that's a multi-step pathway. And so we start out with glucose and then we phosphorylate it and we break it into 2 pieces and so forth. After phosphorylating it, now what we have are intermediates, and we have multi-steps as we go down the pathway to acetyl-CoA and carbon dioxide. And so here's an example of some biosynthesis. You start actually somewhere down in this area, okay? Somewhere in here, maybe at lanosterol, okay? Very common in plants, by the way. So in these arrows here indicate the presence or absence of some kind of a step or a reaction. The numbers here represent specific enzymes. So enzymes will cause, like for instance, enzyme number 4 here, can convert all of these things over to these final products. I'm not going to test you on a figure like this, but hopefully by the time we're done, you'll be able to understand what it means to go from here to say here. that we start out with a reactant. We have an enzyme that lowers the activation energy. Probably have some other reactant going on here as well, and we end up with a product. That basic sort of one-stepper, if you will. And along the lines of the previous figure, the thing I would show you here is that enzymes are the glue of metabolism. And it's amazing how, we tend to take a reaction, we break it out. For instance, we have, let's see, I'm trying to find it here. I don't see it in here. Oh yeah, here we start with glucose and we're turning it into acetyl-CoA. Okay, and then we're going to go through the citric acid cycle and we're going to create ATP from this. But we can also create all these other things. They're all connected, and the glue that holds it all together are enzymes. So let's have a cheer for enzymes. They're awesome, and that's where medicine really lies if you're talking about helping patients. Even if you're not going to give a patient drugs, if you're going to have them work out, what you're doing is you're helping alter their enzymes in places like their heart and their blood vessels and their muscles. Yes, you're doing that. We often think of enzymes as this sort of abstract thing where it's going to react with a substrate, it's going to create something like a product, like, or lysozyme, where we add proteins and it breaks it down into amino acids and so forth. And that's fine. But enzymes are really, really important, certainly for making stuff, like building up your muscles, okay, or creating products that we need for other forms of metabolism like ATP and so forth, or breaking them down. And these are all fine from an abstract sense. Not particularly exciting, right? Unless you are a biochemist or you want to study cell biology or something. But enzymes allow for our muscles to contract. They allow for us to make ATP. In the 22 minutes that you've been sitting with me, you've burned a few pounds of ATP already, so you've got to replenish it. Enzymes are important for your thoughts. Without enzymes, we can't move material from the cell body out to the distal end of the axons. We can't sustain gradients for signaling, and that requires ATP and so forth. Certainly the motor proteins that we talked about before use ATP and energy, and that's an enzyme. These motor proteins are actually an enzyme taking the energy stored from ATP and converting it into motion. Certainly the gradients that we control. And these guys we don't always call enzymes. We don't call protein channels enzymes all that much, but they are catalysts. They are allowing things to move across a plasma membrane that wouldn't otherwise move. The same thing's true with carriers, and we can inhibit them. We can inhibit them just like we can inhibit any other protein in the body. Certain anesthetics are very, very good inhibitors of carrier proteins and channels in our plasma membranes, and they all behave like enzymes. And I realize I'm sort of beginning to beat this to death. I have these slides here, so I'm going to kind of speed up a little bit myself. Enzymes, yes, they speed up the rate of chemical reactions by lowering the activation energy of a reaction. So they are a catalyst. And the reason they are considered catalysts is that by the time the reaction is over, they are reverted to their original state. They're generally unchanged. The substrates are the reactants oftentimes, what that enzyme binds to and does with that molecule. In the case of transporters, I would consider the molecules being transported as substrates. We've also got another word called ligand, but I'm going to get to that later when we talk about receptors. We also have sometimes more than one form of enzyme around, either in different individuals, or we have multiple versions of the same enzyme in the cell for need for redundancy. Maybe one enzyme will work in one situation and another will work in another. For instance, you have certain enzymes that might work in your mouth to break down carbohydrates, but those enzymes won't work well in the small intestine. So you release other enzymes that can do the same thing, only they do it in the conditions of the small intestine. And that word is called isozyme. So you have an amylase that's in your mouth. particularly when you're really, really hungry. Have you ever had that first nacho chip at a Mexican restaurant? Tastes so good, right? Well, by the end of the meal, you eat another one of those, and it tastes like cardboard. That's because the enzyme, the amylase, has diminished in your mouth. But we also have a pancreatic amylase. Does the same thing, but under different conditions. It's a little more acid. It's the pH is different in the small intestine. But enzymes lower the amount of activation energy, and that's how they speed up chemical reactions. They make those reactions more spontaneous, more sensitive to ambient temperatures in the environment. So we control the enzymes. If enzymes control the reactions, in physiology, we're going to control and regulate those enzymes so that we can control the reactions. This is an enzyme right here. This is a really, really cool one. This is a whole family of these. And if you look in the middle here, there's an ATP molecule in there. This protein, and it is a protein, converts ATP, the gamma phosphate, into light. It is a fluorescent protein. So if you see a lightning bug out in the grass in the summertime, you're seeing this protein do its thing. It's hydrolyzing ATP and creating light. And here's another, this is actually a protein complex here. You can see that, see these red things inside and the blue things inside the molecule here as it spins around? Those are an example of some place where we might be able to control this. When I say we, I mean cells and the body can control the rate of this, of how this protein catalyzes the reaction by modifying these areas inside this protein. We're going to talk about that. We're going to break some of this stuff out. And for our final slide for this part one, I want to show you a protein that I was working with. Actually, I was working with both of these proteins. This one in yellow here is a protein called MUNC18. And this protein down here, though it's shown in its non-space filling form, it's just shown in its ribbon and stick figure, is called syntaxin, and these proteins are interacting together. Syntaxin is involved in diabetes synthesis. It is an enzyme. It helps the vesicles that contain insulin merge with the plasma membrane. If you just have a vesicle inside a cell, it will come up to the plasma membrane, it will literally bounce off of it. It's energetically unfavorable for those vesicles to merge with the plasma membrane on their own. So you have this whole network of proteins, and syntaxin 4 is one of them, that helps pull that vesicle down and forces it to merge with that plasma membrane by physical force using ATP. Well, MUNC controls that protein a little bit, and it can interact with it, but we also can control MUNC. You see these sites right here? I created these in a different color. These were all areas that we thought that could be modified chemically, and that could control the ability of MUNC to bind to syntaxin 4 by adding or subtracting groups on these side chains in the protein. And these are side chains of amino acids. We can change the shape of MUNC, and if we change the shape of MUNC, we change how it interacts with syntaxin 4 and how much insulin ultimately is released by a pancreatic cell. So this is where we're going to go in the next part. We're going to show you how we control the controllers. So I'm going to wrap up this video, and I will talk to you in part 2 of chapter 4. Thanks for your time.